Hi there, welcome to the evolution simulation. Here you will learn about the basic concepts of evolution and natural selection. We'll also look at other genetic selection processes to see how they differ. Just so you know, my lab assistant has created a minor emergency in another room and we'll need to take care of it before we start the lab. By the way, how do you feel about dogs? Brilliant. You might be just the person I need. Oh dear, here they go again. I'm embarrassed to ask for help, being an all-powerful AI, but I'm in something of a pickle. My lab technician is on holiday and asked if I could dog sit for them. I thought it would be easy. How do I make them stop? Thank goodness for that. I searched every publication on canine genetics and psychology for an answer, but none of it applied to this situation. I learned so much trying to figure out how to stop the barking. Do you know much about dogs? Pop quiz, which dog in front of you do you think is a different species to the others? Click on the dog you think is from a different species to the others. You got me, that was a trick question. Despite being part of different breeds, these dogs are all the same species, Canis lupus familiaris. Common gray wolves are also part of this species, as mated domestic dogs and gray wolves can produce fertile offspring. Dog breeds are the result of human-controlled selective breeding over the past 10,000 years. The same mechanism created modern corn cobs from ancestors which had relatively few kernels per head. What do we call this trait-conserving process? That's right, we call the process of incremental adaptation in the wild, natural selection. Random mutations can improve or reduce a creature's likelihood of passing their genes to the next generation. In artificial selection, an organism's genetic succession is actively controlled. Organisms carrying strong desirable traits like greater food production, efficiency, or perceived cuteness are allowed to proliferate. Dogs, wolves, and coyotes are all descendants of an ancient wolf-like species. Even further back in history, there's an ancestor that relates to foxes as well. The family tree is so interesting. Come to think of it, this fits nicely with what we're working on today. Let's figure out how we got to have so many different canids running around the planet. Now the pups are quiet, let's head back to the lab. I've left a clone of myself with the dogs, so they aren't alone and unsupervised. I hope they stay calm without you. Okay, let's get back on track with evolution. Fossil and modern DNA studies allow researchers to compare genetic markers between modern and often extinct, ancestral, species. Using such genetic methods to build lineage, trees, is a type of taxonomic research called phylogenetics. Taxonomy is a branch of science that uses different sets of descriptive rules and defining features to group different organisms. In theory, all terrestrial life can be placed somewhere in one taxonomic tree. A common ancestor is an ancient species that links two other species. The offspring of that ancient creature may have migrated or become separated from their original clan. Over thousands of generations, the genes became so different from the original organism genome that mating the ancestral and relatively modern creatures wouldn't produce fertile offspring. An ability to make reproductively viable offspring is one indicator that organisms are of the same species. I was wondering if you can build a basic taxonomic tree of life by linking definitive characteristics and example organisms to their respective kingdoms. Are you up for the challenge? Head over to the card table. The cards on the table represent different characteristics of living organisms. Before you get started, lower the lab pad and take a moment to explore the cards on the table and screen. Hovering over them reveals the defining characteristic they represent. The aim is to place the cards in the correct place on the screen. When you're ready to start, click Continue. Starting with the prokaryotes, associate the characteristics to each domain and kingdom. Great, find the missing characteristic for the Archaea kingdom next. Well done. What characteristic is common to the entire eukaryotic domain? What is the feature common to plants, fungi and animals but excludes the protist kingdom? Nice. Next, add the missing defining features for the plantae kingdom. Almost there. Why are fungi their own kingdom? Finally, complete the tree with the last 2 cards describing the Animalia kingdom. Let's see if you understood how those trees work. According to the image below, which kingdoms have the most recent common ancestor? You've got it. The current hypothesis is that the 2 kingdoms of Animalia and fungi split more recently than the others. Humans, as animals, share a more recent common ancestor with fungi than plants. In fact, the common ancestor shared between fungi, animals, and plants is the same. You're making great progress. You've now reconstructed the basic branches of the tree of life. The detailed outer limbs in taxonomic trees are constantly changing as new data emerges and is integrated. To show how awkward it can be, try placing these species in the correct kingdoms. Put the 3 species cards next to their correct kingdoms on the tree. Correct. As you discovered, some organisms share characteristics across kingdoms, like how the Venus flytrap is carnivorous despite being a plant. Such contradictions give taxonomists headaches. Similarly, spontaneous genetic transfer between species confounds phylogenetic studies, like how ancient bacteria transferred genes into the sweet potato. Place the virus card where you think it belongs in the tree. Sorry, I couldn't resist another tricky question. There's no possible correct answer right now. Viruses are very ancient and adapt to their environment, but controversy surrounds whether or not they're alive. Non-living entities have no place in a tree of life. Alright, it seems you've mastered the idea of taxonomic trees and the evolution of species from common ancestors. Reflecting on my lab assistant's dogs, although they're all the same species, selective breeding has augmented various morphological differences. Breeds within a species, like dogs or corn, aren't recognized as distinct subspecies. We reserve this classification for populations that are geographically or otherwise isolated from one another, like the Bengal, Siberian, and Sumatran tiger populations. Although it seems that evolution makes big jumps, the reality is that evolution progresses in random tiny micro-steps. These accumulate over millions of years to create and fine-tune proliferation advantages. Let's play with idea using a hollow suite of ancient canids, the ancestors of our pet dogs. Move to the second room and take a look at the hollow table. Check it out, tiny canids. This hologram visualizes data from an ancient ancestral colony of early canids. Right now we're observing them at 1,750,000 years before the present. We monitored everything about the creatures, from their feeding habits to the genetic makeup of every member. As we're in a virtual lab, we can holographically model the colony and jump through time to visualize genetic impacts on the colony over millennia. Take a moment to explore the terrain and check for organisms. Explore the hollow terrain and find the canids. Did you find all the creatures? There are 6. The forest provides great camouflage. Let's call this ancestral colony of canids, Generation 0, or G0, for short. I've displayed some frequency data for a random gene on the floating console next to you. I'll call it Gene 3. You can see that each individual has 2 copies, or alleles, for each gene. That makes sense as gene-storing chromosomes come in pairs. One set comes from the mother and the other from the father. The gene is well conserved in this colony. How frequently each allele occurs in the canid population is displayed on the floating console. Usually, we express allele frequency as a percentage of the total number of alleles in a population. You can see Gene 3 has 2 versions or alleles, denoted by the icons carrying 1 or 2 triangles. You can tell if wolves are homozygous or heterozygous by checking the alleles under their image. I bet you can hypothesize the impact of different genetic alterations after a few rounds. Take a look at the allelic distribution data for Gene 3 on the floating console. Let's think about this. If we assume the canid colony has 1,200 members and all members are included in this data set, how many copies of the less common allele are present in the whole population? Right on. You remembered that each individual has 2 copies of the gene, so the total number of alleles in the whole colony is 2,400, not 1,200. 35% of 2,400 is 840, so we can assume that in this canid colony, the less common gene 3 allele appears 840 times, sometimes as a homozygous pair, other times heterozygously. Spin the gene wheel for a random genetic impact. Well, that's unlikely to make much difference. Let's see if the population changes over time without any mutations. Trigger a time jump using the button on the hollow table and check for colony differences. The data console isn't showing any change in the gene 3 allele frequencies. Have you heard of something called the Hardy-Weinberg Principle? It's a tricky concept. In essence, the Hardy-Weinberg Principle states that with no external pressure from the environment and in a system where random mating of individuals is the norm, the genetic balance of a population will stay the same. In a dataset, the principle would mean that having stable allelic frequencies indicates a population in perfect equilibrium with its environment. According to Hardy-Weinberg Theory, what would it mean if the distribution of allelic frequencies within a population shifted over time? Well critiqued. The Hardy-Weinberg Principle supports this idea. Allele frequency distribution would only stay constant in the complete absence of selective pressure. In reality, this situation rarely exists if at all. The environment and the organisms within it are constantly changing. Which reminds me, let's give the wheel a spin and see if a gene mutates in our canids this time. Spin the wheel to induce a random genetic impact. Now we're talking. You got gene 5. Let's check in on the animals and see what effect this mutation has. See if you can spot any differences in the terrain and on the console. I've updated the console with gene 5 data from both the ancestral G0 creatures and the genetically impacted colony we're currently looking at so we can compare them. Check out the canid colony on the hollow table and the data on the screen. Our spinner simulates the random nature of the mutation. Remember, mutations in nature aren't actively induced to make an individual better suited to its environment. Did you notice any changes in the current generation of canids? As we express allele frequency as a percentage of all the alleles in a population, it's only mathematically right that the other would drop. Frequency describes the proportion of alleles within a group that are of a particular type. What do you think will happen to the new allele after a few hundred rounds of random mating? Okay, let's jump through time and see what happens. Trigger another time jump to look for changes in the terrain and data console. It looks like the gene 5 mutation has been removed from the colony. This often happens to uncommon neutral variations. Where multiple neutral alleles of a gene exist, genetic drift generally results in one version becoming dominant and all others being lost. Let's spin that wheel one more time and induce another mutation in the canids. Spin the gene wheel to make a random genetic impact. Awesome, you got gene 9. Let's explore its impact on the colony. Check out the canids and their data. What did you discover about gene 9? Yes, you're right, a new gene 9 allele has appeared. From the data displayed on the console, it already has a 10% frequency in the colony, meaning it qualifies as a genetic polymorphism. There must be some advantage or evolutionary pressure that's keeping this allele around. There is a creature on the screen that is brown but appears to carry a single new variant allele. The ancestral brown allele must be the dominant one, while the new polymorphism is recessive. 2 heterozygous canids could still produce a gray pup. Before we jump, what do you think will happen to the new gene 9 allele in 50000 years? Let's jump time and find out. Trigger a time jump using the button on the hollow table and check for colony differences. That's weird. We can see from the data console that the new allele is still present in the colony, but there are no gray canids in the colony. I wonder what's happened. Nice thinking. If the gene is following Hardy-Weinberg distribution, we would expect more heterozygotes than homologous grays, but some grays should still be present. Our data shows at least one gray is still around. Maybe migration did occur. I'm picking up weak colony life signals in an unexpected and previously unmapped location. Hold on, let me triangulate it. Hooray, we found them. Looks like a small contingent of the colony broke out on their own into this snowy cold terrain. In this new environment, what do you think will happen if we venture into the future? I can see that. Maybe the fur color provides better camouflage. Let's find out. Trigger a time jump to see if any changes occur. Well, would you look at that? The gray fur color must have been an advantage after all. The gray canids have proliferated most successfully and formed a new colony of their own in this new environment. Migration to new environments that favor different genotypes is one of the strongest drivers of speciation. While these canids don't represent a new species yet, they could in theory become one should the colony become isolated from their ancestral counterparts and undergo further adaptation. Speciation takes a solid million years, and we're currently 16,000-ish years in the past, so maybe in the year 984,000 we'll have a whole new canid species to investigate. The incremental changes we've considered in the canid colony are the building blocks of evolution. Speciation can occur in many different ways. What is the difference between genetic drift and natural selection? That's right. These canids result from a change in the climate. Right now Earth is in an ice age, however, the climate will warm in the future. What do you think would happen when the climate changes? I could totally see selective pressure shifting to favor the ancestral allele again if the colony's habitat reverted to temperate forest. Such reversals of natural selection happen all the time, for example when a previously lost apex predator is reintroduced to an environment. Oh my, the clone I left with the dogs only has a short battery. I've enjoyed guiding your evolutionary exploration so much I completely forgot about it. Let's quickly go check that they're okay. Ah, lovely. It seems that these canids are experiencing zero external pressure right now. I'm so glad they didn't start barking at me again. Doesn't it seem wild that all terrestrial life came from one common ancestor? Such variety in only a few billion years. Humans can play the part of nature by actively selecting which organisms to cross, but it's important to remember that natural selection has random mutations at its foundation. The selective pressure comes from how well adapted the life forms just so happen to be to their environment. My assistant must be back. I hope you enjoyed your explorations today. Remember to keep an eye out in your own environment for natural and artificial selective pressures. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.